Well, good morning, everyone. On behalf of the Arthritis Program and my co-chair, Dr. Robert Inman, welcome to the Arthritis Program web seminar series. Thank you all for joining us today. This web seminar series will cover some key topics on various bone and joint-related pathologies and rheumatic diseases. Today is our first lecture. I will have a second lecture, which is going to be on COVID-19 and arthritis. This is going to be on the 26th of May. Our third lecture is going to be on current landscape on therapeutics and scleroderma disease. This is going to be on June 2nd. You'll be getting notification and the calendar invites for those. In the audience, we have members of the Arthritis Program, our key academic institutions, our collaborating partners, our academic advisory board members, president of the Osteoarthritis Research Society International, the ORC, also trainees and investigators, some of our key collaborators around Bone and Joint Institute in London, Ontario, Western University, as well as some of the colleagues and collaborators around Ireland, UK, Sweden, and US. Welcome, everyone. Today's lecture will be delivered by our guest speaker, Dr. Richard Loeser. He's going to be talking about aging mechanisms associated with joint diseases, and especially talking about osteoarthritis and how aging mechanisms are associated in tissue destruction in OA. Dr. Loeser does not need much introduction. He is one of the heavyweights in the field of osteoarthritis, specifically looking at endogenous mechanisms associated with various forms of joint pathologies, especially osteoarthritis. He is the Herman and Louis Distinguished Professor at the Division of Rheumatology, Allergy, and Immunology, and the Director of Thurston Arthritis Research Center at the University of North Carolina at Chapel Hill. And Dr. Loeser has published over 150 research articles. He's the co-editor of the Arthritis and Rheumatology Journal, as is a recipient of various awards, including the Basic Science Research Award from the ORC, as well as research awards from the NIH. So welcome, Dr. Richard Loeser. We're excited to hear to your lecture. One thing which I would like to tell the audience is that you won't be able to ask questions to Dr. Loeser verbally, but you'll be able to type in your questions in the question answer tab in your Zoom link, and we as moderators, Dr. Inman and myself, will read out these questions to Dr. Loeser. All over to you, Dr. Loeser.
Okay, thank you very much. And thank you, Dr. Kapoor and Inman, for organizing this seminar series and giving me the honor to be your first speaker. So hopefully I can give you an informative talk. I know this is a mixed audience of clinicians and researchers, so I've tried to include some of both areas so that hopefully there'll be something of interest to everyone. So just a few disclosures. The only thing on this list that I'll be mentioning are Senolytics from Unity Biotech. Otherwise, no conflicts. So I want to have a brief introduction to osteoarthritis and briefly review the current management of OA for the clinicians in particular, and then talk about the biology of OA, and the focus will be, of course, on the contributions of aging. And I'll discuss some selected mechanisms related to aging in OA, both cell senescence and oxidative stress, and then we'll finish with some conclusions and discussion. So as I think most of you probably understand, osteoarthritis is the most common form of arthritis, affects over 250 million worldwide, and that's probably an underestimate. About 10% of those over the age of 60 will have osteoarthritis of some form. And with the aging of the baby boomers and the obesity epidemic, since age and obesity are key risk factors for OA, the numbers are expected to double over the next 20 years. So this is an extremely important public health problem. And knee osteoarthritis, where you'll see a lot of the research is taking place, is responsible for 85% of the global pain and disability related to OA. And then there's been a steady rise in the number of joint replacements. And I think in large part, this is because we do not have any drugs or interventions that actually target the disease process nothing that's able to slow progression in osteoarthritis. So treatments are symptomatic that I'll talk about in a second. And if we're going to reduce the need for knee replacement, we really are going to have to develop new disease-modifying therapies. So there are a number of risk factors for osteoarthritis, and you can think of these as systemic factors that are shown over here in blue, and then biomechanical factors that are shown in green. So you can see systemic factors: age, female gender, ethnic characteristics, genetics, nutritional factors, metabolic factors. All of these influence your susceptibility to developing osteoarthritis. And we know osteoarthritis has a key biomechanical component as well. Obesity is involved in that, but it's not just obesity increasing the load placed on the joint, but you can think of obesity and its metabolic effects and systemic inflammation that are also important. And so there are two ways in which obesity influences osteoarthritis. Joint injuries and certain types of overuse are important factors. Malalignment, particularly in knee OA, and then changes in joint shape, deformities, particularly in hip OA, elevated bone density, and muscle weakness. So all of these influence biomechanical properties that affect the site and severity of osteoarthritis. So when you look at these risk factors, I think one thing that should come to mind is that osteoarthritis is a very heterogeneous disease. And depending on the individual with OA, one or more of these risk factors may be present. And we'll talk about whether or not each of these risk factors leads to OA in the same way. So just some data to show the relationship between age and OA, and then how obesity can also impact that. So you see here, this is data from Elena Lucena on the prevalence of symptomatic knee OA. And you can see how there's sort of a steady rise here. But then after you get around 50s or so, there's a big jump in the prevalence of knee osteoarthritis. So you can clearly see the effects of age, but also the effects of obesity and gender. So the group with the highest prevalence are females who are obese, followed by obese males, and then the non-obese groups down here. So these multiple risk factors for osteoarthritis appear to be, working in sync in terms of increasing prevalence of OA. So in terms of current management, I'm just going to briefly, for the clinicians, go through the recent our American College of Rheumatology Arthritis Foundation guidelines. And these were published online earlier this year. This was the group of authors that were involved. There may be some from your particular institution. And there are a number of different guidelines out there, different groups, ORSI and so on. But just in the interest of time, I'm going to go through these. And when they put these together and published them, they tried to make it clinically relevant by having strong recommendations for use. And these are modalities that have really compelling evidence for efficacy and benefits that outweigh the harms and burdens. And then conditional recommendations, and these are where the quality of evidence is not quite as good, and there may be more of a balance between benefit and harm. And then they have interventions that they recommend against using, and we'll talk about those just briefly. So in terms of physical, psychosocial, mind-body approaches, you can see those that were strongly recommended, and these are for hand, knee, and hip OA. Exercise, which has been well documented to be of benefit, self-management programs, weight loss, Tai Chi, use of cane, and knee bracing in particular for knee OA, and hand braces for first CMC OA are the ones that were strongly recommended. And then you can see a number of those that were conditionally recommended. Some help, but certainly not harmful. And I'll refer you to the article to sort of spend more time looking through these. And then for pharmacological approaches, strongly recommended oral NSAIDs, which are the most commonly used symptomatic treatment for osteoarthritis, topical NSAIDs for knee OA, still recommending intra-articular steroids for knee, and guided injection for hip OA. And then conditionally recommended some of the other symptomatic treatments that are often used. But then there are a number of approaches that were recommended not to be used. So in terms of the physical, psychosocial, mind-body approaches, TENS strongly against, where harm is thought to outweigh risk, and there's very little benefit from TENS. And then you can see a number of other interventions that are conditionally against. In part, they don't have as much benefit, but the harm is probably not as great. And then the pharmacological approaches that are shown here, strongly against a number of things that have been tested as potential disease-modifying drugs in osteoarthritis. Bisphosphonates, glucosamine, hydroxychloroquine, methotrexate, TNF inhibitors, and so on, platelet-rich plasma, stem cell injection, chondroitin for knee and hip OA, and intra-articular HA for hip OA. So I think, you know, you can make arguments for why these are on this list and strongly against, but I think what it shows you that I think is important for this talk is that a number of modalities that we were hoping could potentially be disease modifying so far haven't shown enough benefit and potential risk of side effects. And so at least at this time, they're not being recommended for treatment of osteoarthritis. So clearly, we need a better understanding of mechanisms that lead to OA so that we can develop effective and safe disease-modifying therapies. And something that I won't have time to talk about today, because this could be a whole separate talk, is that there's a growing thought that a precision medicine approach may need to be used for management of people with osteoarthritis. So we may need to target therapies to specific OA phenotypes or subtypes. And now there's a lot of research being done to try to determine what are the phenotypes or subtypes of OA that may respond to one specific intervention over another. So that's a very active area of research that I won't be able to get into, but I think that's going to be important for the future management of OA. So now focusing again on aging, there are a number of systemic factors that occur with aging that could influence the development of OA. So we know with age, there's decreased physical activity, and this contributes to sarcopenia or muscle dysfunction, as well as an increase in fat mass, which influences a pro-inflammatory state, elevated levels of IL-6. Sometimes this is referred to as inflammaging, and IL-6 is a cytokine that's in studies has been most associated with age. There's a decline in growth hormone and sex steroid productions, decline in bone mass and quality, and a decline in proprioception and balance. And all of these factors could influence the development of OA in different ways in terms of muscle, adipokines, which may affect joint tissues, the pro-inflammatory state, which may be influencing OA, loss of growth hormones, sex steroids can affect joint tissue production, bone. We know bone marrow lesions are important, and these may be in part related to changes in bone quality. And then loss of proprioception and balance may affect how the joints are loaded, particularly lower extremity joints. So these are systemic factors that contribute to OA. And then there are also factors at the tissue level. So some of the aging changes at the tissue level, starting at the top, accumulation of senescent cells within joint tissue. And I'm going to talk a little bit more in depth because I think this is an important mechanism, because these senescent cells make inflammatory mediators, cytokines and MMPs, through something called the senescent secretory phenotype. So we'll talk about that and how that contributes to OA. And then we'll also talk some about oxidative stress, which can affect susceptibility of the tissue to cell death, catabolic activity, and inhibit matrix synthesis. And then the other areas that we won't have time to talk about that are important are the changes in growth factors that I mentioned a second ago, which may affect matrix synthesis. There is, with aging in tissues such as the joint, particularly within collagen, an accumulation of advanced glycation end products. And this affects the mechanical properties of joint tissues, particularly cartilage, making it more brittle, more susceptible to fatigue failure, and this may be an important contribution to aging in OA. Aggregate size decreases with age. There's decreased hydration and increased collagen cleavage, which also affect the mechanical properties of joint tissues such as cartilage. And then there's some evidence that there's increased matrix calcification with aging that could also affect mechanical properties. So again, you can see a number of different things that change with age that at the tissue level also can contribute to osteoarthritis. So to demonstrate some of this, these are pictures that Martin Lotz provided for a review that we made. At the top here, these are femoral condyles from tissue donors, young, normal, healthy 40-year-old, nice, smooth appearing cartilage. And this is a histological section from the same joint. And you can see nice, thick, normal cartilage. And this is stained with safranose that gives it this nice red-pink color, normal underlying bone. Old normal, the tissue looks normal, but it's a little bit brown. And this is from the accumulation of age-related products. And you can see there's thinning of the articular cartilage as shown here. That's the main age-related change. Contrast that with osteoarthritis. Here's osteophyte that's formed here, cartilage loss, and histologically, again, you can see cleft formation, fibrillation, loss of matrix staining, and thickening of the subchondral bone. So the point here is that osteoarthritis and normal aging are not one and the same. They're different processes. It's that aging contributes to the development of OA, but it's not just that your joints are getting old, and that's why you have OA. So as I mentioned, there are a number of systemic factors, biomechanical factors. You can see the changes in the cartilage and the bone that we talked about. It's also important to recognize that the other joint tissues are involved. So meniscal damage in the knee, ligament tears that are very common, and then some level of synovitis also is present in osteoarthritis. And these tissues, as well as the cartilage and the bone, produce a whole host of cytokines, chemokines, growth factors, MMPs that contribute to joint tissue destruction and osteoarthritis. And factors specifically made by the subchondral bone appear to play a role. And as you can see, because of the changes in the bone and changes in the cartilage, how these factors might be influencing the overlying tissue. So this shows you what a complex biological problem OA really is. It's not just simply wear and tear. There's a lot of biology, and it's really a condition that affects the joint as a whole. So as I mentioned, there are a number of different risk factors for osteoarthritis. When you look at end-stage OA, it looks pretty much the same no matter what risk factors people have. But what we're starting to learn is that the pathways that these various risk factors take to lead to OA may not be one and the same. And this suggests that we may need to target different things depending on whether or not this is predominantly aging in somebody or obesity or joint injury or so on. So some evidence for this. We published one study looking at a cytokine called macrophage migration inhibitory factor, or MIF. And this particular cytokine has been shown to be associated with aging. And we looked at the severity of age-related osteoarthritis in mice that had MIF knockout. And you can see both at 12 months of age and 22 months of age, the articular cartilage structure score is a score for cartilage damage, how that is increased in the wild-type mice and significantly reduced in the MIF knockouts, and the same at 22 months. And this is an osteophyte score, again, significantly reduced in the MIF knockouts at both ages. So the MIF knockouts were protected from age-related OA. But then when we took younger mice and did an injury model, and this is the DMM model, destabilized medial meniscus, which is the most common surgical model of OA used for mice, there was no benefit of MIF knockout at all. So here are the wild-type mice, the DMM mice, and the sham, and the MIF knockouts, DMM and sham, no difference at all. And this is a safranose score for cartilage damage, osteophyte score. So it shows you that in terms of MIF, it's playing a role in age-related OA, but at least in this particular model, doesn't seem to be important for injury-induced OA. Something else for the researchers that are using mouse models that I really want to point out that I think is important is that it's very, very critical to consider the age of mice that you're using in your OA studies. So looking at mouse months of age up here and trying to correlate that with human age down here, what you can see that a 3-month-old mouse down here is really a pretty young mouse. And a lot of people are using mice that are 8 weeks old, for example, for OA studies. Those mice would be like studying somebody who's less than 10 years old. And we don't think that that really is where osteoarthritis starts. And so when you look at the mature mouse, those mice are around 12 months of age, and that's equivalent to closer to a 40-year human. So again, think about the age of the mice in your OA studies. And this is another way of looking at it. This is from Jack's lab, where again, they show mature adult mice being a minimum of 3 months of age to 6 months, middle age 10 to 14, and then old mice are between 18 and 24 months. So I just want to emphasize that this is important to consider when you're doing osteoarthritis studies. And a lot of work has been done on immature mice and developing targets for OA, and so far none of that has translated. So age may be important in that. And just to show you a little bit more data to make this point, this was a study we did using microarray to look at gene expression in young and old mice after DMM surgery. And the young mice here were 3 months old, so they were mature, but the older mice were more middle-aged adult mice, 12 months old. And when we looked at the gene expression, only 55 genes were similarly expressed between the young and old mice with DMM surgery, and 493 were differentially expressed. So if you're looking at gene expression and trying to look for new targets, clearly the age of the mice that you're studying are going to have a significant influence on what genes show up in your study. So now to talk a little bit more about aging in more detail. This was a very nice article published in Cell in 2013 that a group of Gero scientists came up with the hallmarks of aging. And when you go around this wheel, you can see altered intracellular communication, genomic instability, telomere attrition, epigenetic changes, loss of proteostasis, dysregulated nutrient sensing, mitochondrial dysfunction, and cell senescence and stem cell exhaustion. All of these are key hallmarks of aging. All of these have been studied in one way or another in osteoarthritis and appear to play a role in how age contributes to OA. I'm going to focus on cell senescence and mitochondrial dysfunction today, but I think any of these are important factors to consider. So in terms of cell senescence, classically, the old way of looking at senescence was replicative senescence, where cells divide over time, replicate themselves, and then stop dividing, and those cells were considered senescent. For a tissue like cartilage and other joint tissues where cell division is pretty rare, replicative senescence is probably not relevant, but there's something called stress-induced senescence. And this can be from both excessive mechanical loading, and we know that's an important factor in OA, and then also mitochondrial dysfunction and oxidative stress are two other factors that can influence the development of senescence. And then there are a number of markers for senescent cells, dysfunctional telomeres, heterochromatin formation, and then changes in expression of particular factors, and I'll focus a bit on P16, which is a transcription factor that goes up in cell senescence that seems to be one of the best markers for senescent cells. And what's becoming really important in this field is that senescent cells have a specific phenotype that's called the senescence-associated secretory phenotype. And this is characterized by the production of a whole host of cytokines, chemokines, MMPs, and particular growth factors and binding proteins. And what's of interest is that there's a big overlap between the factors that are produced by senescent cells in a variety of tissues and the factors that we think are contributing to the development of osteoarthritis. So the production of these inflammatory mediators and matrix-degrading enzymes by senescent cells could be playing a very important role in the development of OA. So next, to just talk a little bit about P16. This is a study from our group here at UNC, led by Brian Diekman, one of our faculty members. This study, we took chondrocytes from human tissue donors of various ages and measured the expression of P16 as a marker. And you can see that there's an increase in P16 expression with age. So this is a biomarker that shows that chondrocytes do age. But in this particular study, P16 knockout mice were not protected from osteoarthritis. So P16 is a good biomarker, but it may not be necessary for the development of OA. As I mentioned, there are a number of other changes in addition to senescence and oxidative stress that have been published on. And I don't have time to go through all of these, but some that are really important, I think, is that there's a reduction in autophagy. And this reduction in autophagy seems to affect matrix synthesis and degradation and cell death. The mitochondrial ROS I will talk about, but changes in SIRT1, AMP kinase are also important. Changes in the response to growth factors such as TGF-beta, which in part may be due to changes in these receptors, the ALK1 to ALK5 ratio that changes with age, and then a number of nuclear factors, HMGB2, and the FOXOs, which Martin Lotz's group has done a lot of work on, are important aging components as well. So our particular interest in our lab has been to look at the role of oxidative stress in aging and its contribution to OA. And I'll give you some evidence for how ROS increase with OA and their role in development of OA. Biomechanical stress is also a factor that, in addition to aging, can result in increased production of ROS. So two of the OA-related factors, aging and biomechanical stress, are both important here. And then in a series of studies, we found that ROS, intracellular ROS, inhibit anabolic signaling, for example, IGF-1, and promote catabolic signaling, such as through cytokines and matrix breakdown products like fibronectin fragments. And this reduces growth factor production and matrix synthesis and elevates productions of MMPs and cytokines. And in this way, you can contribute to the development of OA. And then I mentioned the age formation, which I don't have time to talk about it, but again, affecting the biomechanical features of the tissue that can also contribute to this. So, like I said, there's lots of evidence that reactive oxygen species can contribute to OA. There are studies showing certain antioxidants are reduced in aging in OA. There's a reduction in superoxide dismutase 2, which is in the mitochondria, particularly its activity goes down with age. Glutathione, catalase, which is an antioxidant enzyme, extracellular superoxide dismutase, and then NRF2, which is a regulator of antioxidant gene expression. And then there's evidence for increased levels of oxidative damage, nitrotyrosine, lipid peroxidation, and so on. And then mitochondrial dysfunction is important to all of this, because with mitochondrial dysfunction, you have increased production of reactive oxygen species. What's also important to recognize is that you need a normal level of reactive oxygen species for normal homeostasis. So there's a redox balance controlled by the level of ROS production and the level of antioxidants. Oxidative stress is where you have an imbalance in these two sides, where there are excessive reactive oxygen species relative to the antioxidant capacity of the cell. And this redox imbalance causes oxidative damage, which has been well characterized. But what the field is starting to really recognize is that it's not just simple oxidative damage, but it's changes in signaling, disrupted cell signaling, that's important. And this is probably one of the more important mechanisms by which oxidative stress contributes to age-related diseases. So some evidence for this, we published one paper where we looked at a marker of age-related oxidative stress, and that's hyperoxidation of this protein called peroxyredoxins. And these are the key antioxidant proteins within cells. When they're hyperoxidized in this SO2, SO3 state, they are inactivated, and so they can no longer act as an antioxidant. So we found with OA and aging that there's an increase in the inactivation of these important antioxidants, and this affected cell signaling. So it reduced pro-survival signaling and signaling important in matrix synthesis. That's just a higher power view of the PRX3 SO2 SO3. And then in another study, important signaling protein, JUNK, or C-Jun N-terminal kinase, it's known to be activated by low levels of reactive oxygen species. And this is just phosphorylation of JUNK, and then its downstream substrate C-Jun. But as you can see, when you get to higher levels of ROS, JUNK is actually inhibited. And so again, this shows this redox balance where you have sort of physiologic signaling that may be occurring at lower levels of ROS, and then when you get to elevated levels of ROS, you inhibit certain signaling proteins. Initially, you would think, well, this could be good because JUNK is a MAP kinase that's been implicated in cartilage destruction and osteoarthritis. But we looked at that, and in JUNK knockout mice, we found that those that had JUNK knockout, either JUNK1 or JUNK2, actually have more severe age-related OA. So this oxidative stress inhibition of JUNK may be a contributing factor to increased OA with aging. You can see articular cartilage score and osteophyte score both went up in the JUNK1 and JUNK2 knockout mice. And this is just published recently online in Arthritis and Rheumatology. We also looked at young mice in the DMM models, and the JUNK knockouts were not protected at all in osteoarthritis models. So we wanted to see, well, how could JUNK knockout be contributing to the development of osteoarthritis? So we looked for P16 as a marker for cell senescence, both by immunohistochemistry, which is shown here. These are wild-type control mice and JUNK1 knockouts. The brown cells here are in the synovium. So you can see strong synovial staining for P16 as a marker of senescence in the JUNK knockouts that's not present in the wild-type control. And then this is using immunofluorescence, which is more sensitive. Here we're showing the JUNK2 knockouts. And again, most of the senescent cells are here in the synovium, with fewer in the meniscus and the cartilage. And then this is underlying bone where you see senescent cells. So what we found was with aging, compared to the wild type controls, the JUNK1 and particularly the JUNK2 knockouts had an accumulation of senescent cells. So what it suggests is that JUNK is important for maintaining homeostasis to inhibit cell senescence with aging. So in these JUNK knockouts, you actually get more severe OA rather than less OA, which was what expected. So kind of in summary of what our model of osteoarthritis suggests, and this is backed up by lots of really important work by a number of different labs. Abnormal mechanical loading clearly contributes to osteoarthritis. This causes matrix damage, but also increased production of reactive oxygen species. And then changes that occur with aging, mitochondrial dysfunction, oxidative stress, autophagy, many of the others are also important. And these combine with other OA risk factors. So together, this can start the process of damaging the matrix and increase the production of pro-inflammatory mediators. These matrix fragments can feed back on receptors. These are integrins shown here. Activate signaling pathways that increase ROS production, altering signaling, promoting more catabolic over anabolic, and the increased production of inflammatory mediators. What should normally happen in sort of a chronic wound process is that as you start to damage the matrix, you release growth factors. And growth factors should actually induce a repair response and turn off these pathways. But what seems to happen with aging and OA is the cells are less responsive to growth factors because of some of these factors here. So you tip the balance more towards the catabolic side. You have increased production of inflammatory factors, increased matrix destruction, and then this leads to the development of progressive osteoarthritis. So you can see there are lots of potential places within this model where you might be able to intervene and target something that might slow progression of osteoarthritis. So in conclusion, osteoarthritis is a very complex, heterogeneous condition. It involves a number of inflammatory mediators and matrix-degrading enzymes. Aging changes that are relevant to osteoarthritis include both systemic changes as well as local changes. Cell senescence, oxidative stress, and other aging changes are important. And then I think these senescent cells that produce cytokines, chemokines, and MMPs are going to be an important target for intervention. And these are being targeted by something called senolytics. And so there's preclinical evidence, particularly from Jennifer Aliciev's lab at Hopkins, showing that if you kill senescent cells, at least in a mouse model, you can slow progression of osteoarthritis in a surgical model, but also with aging as well. So this is something that's in early phase studies now that is being looked at as a potential new treatment for OA. I'd also like to recognize my lab group that over the years, this group has made significant contributions to our work, particularly John Collins, who's now a junior faculty member at Jefferson, and Scott Wood and Veronica played important roles in some of the work I've talked about here. Important collaborators that are listed here: Susan Chubinskaya, who gets us tissue, and Kathy Carlson's group, who does histology for us, our tissue sources, and then our funding sources. So I've tried to leave as much time as I could for questions and keep the talk relatively short, because I think the discussion is always the part that people are interested in. So I'll kind of go back to maybe this slide, and then we can open it up for questions.
Great.
Thanks so much, Richard.
That was terrific. Perhaps, Mohit, can I start off with a question for Richard then? I was struck, Richard. A question really is what the current thinking about the subchondral bone contribution is, both in terms of its stiffness and also the MRI demonstrated lesions in the subchondral bone. Because I was struck in your junk knockout mice how much the staining was enhancing the immunofluorescence in the subchondral bone.
Yes, that's a good point. So in terms of the senescence part, I'll answer the last part first. There is evidence that osteocytes are a type of cell that gets senescent with aging and can produce these inflammatory mediators, so that may be playing a role. You know, it's been thought for a long time that the subchondral bone is contributing to osteoarthritis, and there are certainly phenotypes of OA where subchondral bone is important. High bone mass is contributing. You know, whether or not it's stiffness of subchondral bone, that was a theory David Burr and Eric Radin had, that they kind of disproved and thought that it was not just simply the stiffness of subchondral bone that was affecting the overlying tissues, but it may be more of these other mediators that are being produced in bone that are important. There's studies showing TGF-beta, for example, that's made by mesenchymal stem cells in the bone may be important, and so on. So I think it may be a biologic contribution of subchondral bone that's as important as a mechanical contribution. And again, that may play a role more in some people than others. But overall, the point is that all of the tissues are affected in a way. And so in part, they can all contribute. And you mentioned the bone marrow lesions. Those certainly seem to be important, particularly in pain, and they're associated with progression of OA. And those seem to be formed in part by mechanical changes. So mechanical forces on the bones seem to be contributing to where they form, and they could be playing a role as well.
Thank you.
Richard, we have some questions which are being posted by our audience. So first question is by Jason. He's asking, the differences you see in injury versus aged mice, do you think this is the result of what cells or tissues are primarily driving the joint damage? For instance, is cartilage and chondrocytes driving age-related OA versus synovium or inflammatory cells driving injury-induced OA?
You know, it is much more complicated than that, because if you look at our junk knockout mice with more age-related OA, it's actually the synovium where these senescent cells are present. So the senescent cells in synovium producing inflammatory mediators are actually contributing to age-related OA. And so I think it's really, all the tissues can contribute to either type. So there's lots of work on joint injury, at least in preclinical models, knocking out genes in joint tissues such as the cartilage, protecting from OA. But again, depending on your model, the level of synovitis can also be different. And so I think, you know, you have to think when you're looking at preclinical models, which model you're using, how much inflammation is there, what are the age of the animals, and so on. But I think, again, multiple tissues can contribute, and there's not a distinction between which tissue is age-related and which tissue is injury-related.
Thanks, Richard. We have another question by Starley, who's asking, first of all, thank you for shining a light on too often overlooked limitation of many OA studies. Regarding aging, do you think it is the act of aging that contributes to OA, or could it be the decline in activity that results that adults tend to do as they age?
Right. So, yeah, that was sort of a point I tried to make on an earlier slide where I showed the systemic changes with aging, and decline in physical activity was certainly one of them. But then there are all of these other age-related changes at the level of the tissue and cells. So my answer to that question would be, it's both. So clearly the reduction in physical activity and normal physical activity is important for joint health, that's playing a role, but also the age-related changes that are occurring at the level of the tissue and cells. So it's a contribution of all of these factors rather than one single factor overall driving the contributions of age to OA.
A follow-up discussion on this as well, because Starley says that, is there evidence that older people who exercise, so for example, if somebody is doing Tai Chi, counteract age-related process better?
There is, you know, there is not much evidence that exercise by itself slows disease progression. So like in clinical trials, 18-month trials of exercise, you certainly improve pain, you improve function, but it doesn't seem to directly affect progression of the disease by MRI or by biomarkers. So I think that exercise is very important in terms of pain and function, but by itself, there's not evidence that it can actually slow disease progression. You know, people that exercise as they get older are usually healthier people, and they do seem to be at lower risk overall of getting OA. But again, there's multiple factors involved, and so you know, it's certainly something we want to encourage everybody to do, but I don't think you could tell somebody, yes, there's evidence that if you exercise, you're going to prevent yourself from getting OA.
We have a question from Tristan Meritz in the University of Michigan. Thank you for a great talk, Richard. Regarding the senescence finding in the JUNK1 knockout mice, do you have any evidence regarding synovial macrophages versus synovial fibroblasts mediating this?
No, that's something we want to look at next. So we know there are senescent cells lighting up there, but how many of those are fibroblasts, how many of those are macrophages, we haven't looked at yet. We were getting ready to do a lot of this, and then our lab was shut down with COVID-19. So I'd already started talking to our collaborator, Kathy Carlson, her lab was going to start doing some of this. So that'll have to wait. But yes, that's a really important question. Which cells are senescent and contributing to this? You know, there is work by Jennifer Alicia's lab showing senescent cells in synovium in even an injury-induced model, and it seems to be a number of different cell types that are senescent. But we'll be looking at that as well.
Thank you, Richard. We have another question by Dr. Robin Poole. Hi, Richard. Can you discuss the relationship of chondrocyte hypertrophy in OA to senescence?
All right. Hi, Robin. Glad to hear you're on. I'm not surprised by your question. For those in the audience who may not know Dr. Poole, he was one of the investigators who made significant contributions to our understanding of osteoarthritis over the years. And one of the areas that he was most interested in has been chondrocyte hypertrophy. And he described in osteoarthritis increased numbers of hypertrophic cells, and these are characterized by expression of type 10 collagen and also MMP13, which is made by hypertrophic cells. So I do agree with Dr. Poole's studies that chondrocyte hypertrophy plays a role, and certainly some of these aging-related changes are associated with chondrocyte hypertrophy. And so I think that's definitely a considering a factor. It'd be interesting to actually try to look at co-staining with, say, P16 and type 10 collagen to see if all the same cells are hypertrophic and senescent. That hasn't been done yet, but I think that would be an interesting study to do. So thanks for the suggestion.
Somia raises a question, Richard. I'm curious to understand the killing of senescent cells as a possible therapeutic target. We're seeing in other inflammatory conditions apoptosis of cells, and therefore cytosis by macrophages seems to be an important immunomodulatory mechanism. So the question is whether targeting senescent cells could be a therapeutic avenue.
Right. So that is a very good question, because you always are concerned when you start killing cells within a tissue, including the joint, is that really going to be of benefit over time? The thought is that these are the senescent cells that are producing these inflammatory mediators. If you remove them and you leave the other healthy cells alone, that could be of benefit. There is definitely preclinical evidence in mouse models that this works. And in addition to joint tissues, it's being looked at other aging-related tissues as well. And so at least the preclinical models suggest that it's beneficial. Unity Biotech has done a phase 1 study of injecting a senolytic into the joint for safety, and they're starting phase 2 studies. So I think we'll eventually know the answer in terms of humans and at least NEOA. And there are a number of different companies looking at this. And like I said, a number of different other diseases where people are thinking that killing senescent cells will be of benefit. So I think the evidence is that it can be helpful, but there can be a downside. I should say that cell senescence doesn't just happen to be bad and be aging related. Cell senescence has normal physiologic roles. So for example, wound healing involves cell senescence, and the killing of senescent cells could potentially slow wound healing. And that's one reason why it might be better to have an intra-articular therapy, at least for OA, rather than a systemic therapy. And then cell senescence is also a way in which our bodies reduce malignancy. So for example, P16, which is expressed by senescent cells, decreases replicative capacity. Mutations in P16 that inhibit its function are associated with malignancies. So there's this balance of malignancy, cell senescence that's also important. So there are important physiologic roles for cell senescence that we also have to keep in mind. And so that's why this is going to be, I think, important to study very carefully and look at both potential benefits and harm.
Thank you, Richard. Another question is from Mayoa. As you mentioned, the young mouse model is not suitable for aging-related OA studies. How about iPSC-derived OA models? As known, reprogramming can reset the cell state to bring them back to the embryonic stage. does this mean iPSC model is less suitable for studying age-related OA studies?
Well, I think that for cell culture models, yeah, sure, that can be useful. And I think we learn a lot by doing cell culture studies. But at some point, you're going to have to go in vivo, because there are lots of limitations to studying things in culture with primary cells, with iPS cells, and so on. So I think, depending on your study, depending on the questions you want to ask, sure, there are different ways that you can study things in vitro, but it's still going to come down to showing that these same processes are happening in vivo. So J and K is a good example. We had lots of evidence that signaling downstream of J and K was important in vitro. And when you blocked it, you could block production of MMPs and so on. But then when we did the studies in vivo, we did not see what we expected. In fact, we saw the surprising worsening of OA. And so, I think that you have to always keep in mind with your cell model, if you're going to be able to use this to translate it to the in vivo situation, you need to be asking the right questions. And there's probably not one cell model that's superior over the other.
Thank you, Richard. There's actually a follow-up question to the first question, which was asked regarding the chondrocytes in the synovium. Is there any information that JNK signaling or JNK is differentially regulated in chondrocytes versus synovial cells, macrophages, that may allow for specific signaling to promote joint degeneration?
So there could be differences between the two. So in this particular study that we did, the JNK2 knockout was a genomic knockout, so it was in all tissues. Whereas we knocked out JNK1 both globally and also in a tissue-specific manner. And I didn't show the data, but we also had a double knockout of JNK1 and JNK2, which was JNK1 in a tissue-specific manner in joint tissues with aggrecan-Cree in the JNK2 global knockout. At the time we were doing these studies, and I think it's still true, there's no floxed JNK2, so that we couldn't specifically look at JNK2 knockout in a tissue-specific manner. So I guess the short answer is, I don't know for sure, but I can just say that the JNK2 knockout was would have been synovium as well as in the rest of the joint tissue, since it was global. And that's where we saw this increase in senescence.
Thanks, Richard. Next question is from Dr. Mari Yurovitz. With so many inflammatory mediators produced, why do you think that the classic anti-inflammatory drugs such as methotrexate have not been found to be useful?
Well, methotrexate, has some effects on inflammation, but it's not, like a biologic that inhibits a specific inflammatory mediator like a cytokine. So, clearly methotrexate is useful in treating rheumatoid arthritis, but its effects don't seem to be specifically by inhibiting an inflammatory mediator such as TNF or IL-6 or so on. And that's why biologics, are commonly used when methotrexate's not working. So in OA, I would say that the ability of methotrexate to inhibit the inflammatory mediators that are important in OA is just not sufficient. Although, there's some studies that show a little bit of benefit in methotrexate and some that did not. So there's sort of some conflicting data on methotrexate in OA as well. But I think really for OA, we're going to need some better, more specific targets. And, the point is correct that there are a number of different inflammatory mediators. We just don't have that breakthrough yet, knowing which is the driving factor, like they did years ago in RA, where they found that TNF was a driving factor, at least in a proportion of people, and then found other, mediators like IL-6 and JAK signaling and so on. The field exploded. We're not there yet in OA. inhibition of IL-1 doesn't seem to help in OA. Inhibition of TNF so far hasn't helped. And so, you know, I think so far we're not looking at the right inflammatory mediator, but I can't say we know which one's the key one yet.
One question, Richard, in the event that we do have what might be a very novel therapeutic intervention, what is the patient population and the stage of disease that we should actually be studying? How do we identify the patients at the right stage where there's a potential for reversibility?
Great question. Clearly, if you are treating people at end stage OA where there's complete joint tissue destruction, you know, bone on bone, no matter what intervention you have, short of joint replacement surgery, it's not going to work. So anything that's going to target disease progression has to be done at an early enough stage that you would prevent more severe joint destruction, which is not going to be reversible. And so there's a lot of work being done on the best way to find people at an earlier stage and then determine who's going to be at higher risk for progression. And this is going to include imaging, so lots and lots of work being done to look at MRI and different pulse sequences of MRI that might pick up early OA. And then this phenotyping precision medicine work to determine who is most at risk for progression that would be responding to a particular intervention. That's going to be really key as well. So it's a very active area of research, and lots of groups working on that. And I think that's going to be important for this field to move forward.
Richard, we have a question from Dr. Nigel Haroon. With MIF, small molecule inhibitors, and monoclonal antibodies against CD74, do you see any potential in targeting this pathway for age-related OA?
Possible. You know, we're the only group that I know of that's actually, you know, studied MIF and OA. So it'd be nice to see someone sort of replicate our findings, first of all. You know, one of the issues, I guess, with targeting MIF and age-related OA is sort of, you know, when would you start it, who you would start it in. And so that's related to the previous question. You know, we really need to figure out the best population that might be treated with a MIF inhibitor. So, you know, I think that could have potential. I think more preclinical work would need to be done, particularly with the MIF inhibitor, because this was a global MIF knockout that we studied. So it's got potential, but that definitely needs more work.
Well, Richard, we are at 9 o'clock. We still had one more question left to answer, so we won't be able to read that. But thank you so much for joining us today and taking our time to give this amazing lecture and discussion. And to the audience, please don't forget to join us next week for the lecture on COVID-19 and arthritis. Thank you again, Richard.
All right. Yep, thanks for having me, and thanks for all the great questions.
Absolutely. Thank you, everyone. Okay. See you next week.
Thank you.
Bye.